If possible, very briefly about the oncovaccine, literally 2 words. The whole world is working on the creation of personalized medicines based on the biological codes of a particular person. And this includes a personalized cancer vaccine. Clusters of 4 of our centers, led by the Center for Physicochemical Medicine, have developed a technology for verifying mutations that caused a particular person's tumor, isolating neoantigens that are recognized by the human immune system. And within these, respectively, antigens, the release of those peptide fragments that are just a signal for the immune system of a particular person. The second technology is the synthesis of these selected peptides in such a way that turn on the immune system, direct it to the tumor. And the third point is the creation of vaccines themselves, for which these peptides are the active ingredient. So, we had to make several vaccines on different platforms, on different carriers. These are also peptides on nanoparticles. These are peptides within the framework of a vector vaccine, on an adeno-associated virus. And this is the mRNA of the vaccine. Why is this important? Because the dynamics of the immune system activation is completely different, and for each person one needs a quick response, and the same number of short ones. And to do this, we need to include this answer in a week, in 2, and so that it lasts for 3 years. These are different approaches, as well as different choices of antigens. And as a result, in an experiment with the most malignant intestinal adenocarcinoma in animals, we revealed not just a sharp slowdown in tumor development when the vaccine was administered, but in the event that there is already a tumor, a reduction in its volume by 75 to 80%, and animal survival. In other words, animals do not die from this tumor, they continue to live. There is an opportunity to control it. We have actually completed a 3-year research cycle on this vaccine. Due to the fact that the law on the use of personalized drugs was adopted this year, we hope to start using it in patients from next year. In parallel, and with other localizations of oncological diseases, we conduct studies number 2 and number 3. This is melanoma, skin cancer, and this is a malignant formation of the glioblastic brain. That's the last thing. Vladimir Vladimirovich, I mentioned mRNA vaccines. We managed to make a real breakthrough. We are now the first in the world in this direction. We have created a unique platform for the creation of mRNA vaccines. This is a vaccine that is carried not by the antigen itself, but by a nucleotide code. It enters the cell, and the cell itself gives birth to a protein based on this code, the one that is needed. A platform has been created that allows self-assembly of lipid nanoparticles with a diameter of 200 nanometers. And inside this particle, by immersing RNA with the existing biological code, and outside accordingly, anchor special components that ensure the biocompatibility, stability of this particle, rapid penetration into the cell, and other features. As a result of these few know-how of our scientists, we have a significant advantage over well-known world analogs in a comparative study. This is a modern Pfizer, 2 companies that produce these vaccines. Firstly, in terms of effectiveness, this vaccine, in terms of the efficiency of protein production from the code that is introduced, exceeds the Pfizer by 65% and the modern by 90%. And these are highly reliable things. And the second point, it means that the whole world criticized these 2 vaccines after the COVID pandemic for causing a huge number of side effects of the mRNA vaccine polysystem. We managed to modify the introduction of this and the vaccine market in such a way with the help of special biotechnological techniques that we actually zeroed out the side effects. That is, the vaccine acts only at the injection site and appears immediately in the immune blood cells and produces its effect, without affecting other organs of the system. This platform, it was the basis for the oncovaccine and, when administered to an animal, reduced it by more than 80%.
How much more time is the work calculated before it can be applied? Well, roughly.
Well, the thing is, I think that in order to ensure complete security is guaranteed, we need probably about a year more in order to double-check everything, each component separately and all together.
Well, it's going to be a breakthrough anyway.
We really hope so.
Thank you.
Thank you.